Hello everyone. Today we will learn about nucleic acids. So nucleic acids carry the genetic information because they have been isolated from the nucleus of eukaryotic cells. So there are two related types of nucleic acid, as we all know, DNA and RNA. So DNA stands for deoxyribonucleic acid and RNA is ribonucleic acid. So let's go in detail. So the master copy of each cell's genome is stored on the long molecules of DNA, which may contain many thousands of genes. It is kind of linear molecules, and a gene consists of some segments of DNA. Like this could be a gene, like a first gene, second gene, or third gene, and it is a long segment of DNA. And so each gene is thus a linear segment of a long DNA molecule. And in contrast, RNA molecules are much shorter. They are much shorter and can use to transmit the genetic information to the cell machinery in the form of proteins, and can carry only one or few genes. So DNA and RNA are linear polymers made up of subunits. They are linear polymers made up of subunits known as nucleotides. So the information in each gene is determined by the order of different nucleotides. There are 4 different nucleotides in each type of nucleic acid, and their order determines the genetic information. So RNA has different 4 nucleotides and DNA has different 4 nucleotides. So each nucleotide has 3 components. First one is a phosphate group. Second is a 5 carbon sugar. And third is a nitrogenous base. So the phosphate groups and the sugars forms the backbone of each strand of DNA or RNA. It forms backbone, and the bases are joined to the sugars and stick out on sideways, like we can see in the later diagram. So in DNA, the 5 carbon sugar is in DNA it is deoxyribose, while in RNA it is ribose. Both are 5 carbon sugar, but in deoxyribose there is one less oxygen than ribose. So let's see the diagram of ribose and deoxyribose. So let's see, this is, so these are carbon, 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 okay, and here is CH2. So here we have OH, H, 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 OH, H, OH, and here we would have H and there OH. So this is deoxyribose. Why? Because it does not have oxygen on the second position of carbon. This is first, second, third, fourth, and fifth. Okay. But here on ribose, let's look at the diagram for ribose. We have carbon here, CH2 here, oxygen OH, C, okay, H, OH, H, and OH. Likewise, OH and H. So here in ribose we have oxygen on the second carbon. second carbon. So that's why it is known as deoxyribose, because it lacks oxygen on the second carbon. Okay, so now nucleotides are joined by linking the phosphate on fifth carbon of the ribose of the on one nucleotide to the third position of next nucleotide. Like I can show you how. We have, let's take an example of deoxyribose. So let's say we have this H and H CH2 O H OH and H. So this is deoxyribose. So first, second, third, fourth and fifth. So the base is attached to the first carbon and phosphate is attached to the fifth carbon. So now how it forms the entire structure, we'll see. The phosphate group is joined to the sugar on either side by ester linkages, and the overall structure is therefore a phosphodiester linkage. This is one nucleotide, and then to the other nucleotide we can see how they are linked. So if we have phosphate, let's draw this phosphate as, this might have the negative charge. Okay, now this oxygen is attached to the other nucleotide, the third carbon. So it would attached like this. Is the whole situation, I guess. Okay, so this is the third carbon. This might be the, this is 1 2 3 4, and here would be the CH2. Yeah, this would be the third carbon. So it is attached to the third carbon of the another nucleotide. Phosphate group linking the sugars has a negative charge, so it gives DNA an overall negative charge. So the DNA is always negative. Now coming to the nitrogenous bases. So how these nitrogenous, like what kind of different types of nitrogenous bases are there? So there are 5 different types of nitrogenous bases. Adenine, guanine, cytosine, thymine and uracil. So these bases, okay, so adenine, guanine, cytosine and thymine are found in DNA, but in RNA, instead of thymine we have uracil. So if there is a DNA and it is transcribed to RNA, like we have A, T, G, C, T, A, so then if it has been transcribed to RNA, so it would be U, A, C, G, A, and then again U. So there would not be T. So this is RNA and this is DNA. Okay. Complementary base pairing we will learn later in the next sessions. So it's just an example. So okay, uracil is found in RNA instead of thymine. Just remember this for now. Okay, so these bases found in nucleic acids are of two types. These bases are of two types, purines and pyrimidines. So pyrimidines are smaller, having single ring. They are smaller, they have single ring. while purines are bigger, and they have fused rings, like fused double rings. They have two rings which are fused together. Okay, so adenine and guanine are purines, while cytosine, thymine and uracil are pyrimidines. So now let's look at the structure of these purines and pyrimidines. So the general structure of pyrimidine is N. So this is the general structure. Just remember, this is 1, 2, 3, 4, 5 and 6. It is a hexagon ring, and having the nitrogen at first and third position. There is no carbon at third and first position. It is nitrogen. And except these two positions, the other position 2, 4, 5 and 6 have carbons. Okay, so this is general structure for pyrimidines. The general structure for purine is like this. Start from here. 1, 2, 3, 4, 5, 6, 7, 8, 9. Now remember, so here in purines, the numbering we do is always anti-clockwise, and in pyrimidines, here it is also anti-clockwise, but you can do clockwise also. Then you have to invert the structure. But okay, remember, they have the numbering anti-clockwise, but in purines the nitrogen is present in only odd numbers. The nitrogen is present only odd numbers, like 1, 3, except 5. It has nitrogen on 7 and 9. So nitrogen on 1, 3, because remember from 5 it is joining, 7 and 9. So we have nitrogen on the odd positions. Here also we have nitrogen on the odd positions, but here it has a single ring. So don't be confused with these. The nitrogen is always on the odd position. But in purines it has the numbering till 9, and in pyrimidines it has the numbering till 6. Now coming to the specific structures. So for purines, so first for adenine we have, look at this, C, NH2, again C, here would be N, then we have N, C, then N. Okay, now here we would be having double bond. Okay, now first number this 1, 2, 3, 4, 5 and 6, 7, 8 and 9. Now add double bonds, जैसे कि बेंजीन में होता है वैसे ही add double bonds to it. This would also be having a double bond, and this. Okay, this is the structure for adenine. It has NH2 at 6th position, and then just complete the structure with the remaining hydrogens, just completing the valency of carbon and nitrogen. Okay, completed. So we have adenine, we have nitrogen on first and third position, and an amine group, NH2 group at 6th position, and also nitrogen on 7th and 9th position. Okay, and we have double bonds on just like the resonance structures have the double bonds, just like that, just like benzene has the double bonds. Similarly, we have the double bonds on it. Coming to guanine, so this was adenine. Now coming to guanine, we have C, N, again 1, C, N, 3, C, then N, then C, then N. Complete this structure. Now number it 1, 2, 3, 4, 5 and 6, 7, 8, 9. Now add the double bonds. So first, instead of amine on 6th position, we have double bond O group. Okay, and here we have amine on 2nd position of carbon, 2nd position. So now add double bonds. Here all the valencies are complete, just we have to add H to the N. Here are all the valence. No, we have to add double bond to it. We have to add double bond to it and then double bond. Okay. Now, so instead of amine group at 6th position, it has amine group but it is on 2nd position, and double bond O group at 6th position. Just these two structures are very similar to each other, but then we have to remember that on 6th position adenine has amine group, and on 6th position guanine has double bond O, while its amine group is present on the 2nd position. Okay. So now coming to the structures of pyrimidines, first we have the structure of thymine. Thymine is just a hexagon like this. Okay. Double bond O, NH, double bond O, CH3, CH, NH. Now coming to the numbers, we have 1, 2, 3, 4, 5, 6. Okay. So it has methyl group on 5th position, and double bond O group at 2nd and 4th position. Remember, so thymine has, okay, now adding the remaining double bonds. You can just check if the valencies are complete, and if it's not, just add hydrogen or double bonds to it. Okay. Okay, so thymine, just remember like this: on 1st and 3rd position it has nitrogen, 2nd and 4th position it has double bond O, C double bond O. Okay, and at 5th position it has methyl group. Okay, and 6th position is empty. There is nothing on 6th position. 1st and 3rd nitrogen instead of carbon, 2nd and 4th C double bond O, 5th position methyl group, and 6th has nothing. Then cytosine has has double bond, this does not has double bond, this has this H, and yeah, 1, 2, 3, 4, 5 and 6. This is cytosine. So for cytosine, we can see again 1st and 3rd has nitrogen. Okay, 2nd has C double bond O. Okay, 4th has amine, CH2 group. 5th and 6th has nothing on it. Okay, just remember like this. Now lastly we have uracil. So uracil is easiest, I guess. For me it's easiest. Okay. Yeah, like this: 1, 2, 3, 4, 5 and 6. So look at this: uracil, 1st and 3rd have nitrogen, second and fourth has C double bond O, fifth and sixth has nothing. So just look at the structures more closely and see that thymine and uracil have structural similarities. So that's why thymine is replaced by uracil in RNA. So thymine has this methyl group at 5th position, but uracil does not have this methyl group at 5th position. Otherwise the structure is same. The cytosine has a little bit different structure. It has NH2 amine group at the 4th position instead of the oxygen, while otherwise it has the same. In purines we have adenine. So in adenine we have, okay, again we can just wrap this up like this. Adenine we have 1, 3, 5 and 7 nitrogen, and on 6th position we have amine. Okay, and in guanine 1, 3, 5 and 7 has nitrogen. On 2nd position we have amine, and on 6th position we have double bond O. You can remember like this. And yes, this was all about the structures, all the structures of purines and pyrimidines and the nucleotides. And let's just briefly discuss about nucleotides and nucleosides. So nucleoside, what's the difference between nucleotide and nucleoside? So a base plus sugar, a nitrogenous base plus sugar is nucleoside. It is nucleoside, while base plus sugar plus phosphate group is nucleotide. So nucleotide could be nucleoside plus phosphate. Okay, we will dive deep into the concepts of the structures and the backbone, how the backbone is formed, and how the DNA, the double-stranded DNA forms the double helix. We will discuss in the later videos. If you like this video, just give it a thumbs up, and please subscribe my channel if you are new to my channel. And if you want these notes, I can provide you the link. You can just comment me, I can provide you the link. And thank you so much for watching, and again we will discuss the next part in the later videos. Thank you.